Hello everybody. Bonjour. Selamat pagi. I'm from Indonesia, so I'd like to present it about related to health analysis of Salmonella species contamination on unwrapped and wrapped food in Darul Islamic Boarding School, Jombang. So start from the introduction. So the typhoid fever is an infection disease caused by Salmonella typhi or paratyphi. And Salmonella is transmitted through contaminated food. And then this infection is considered as a serious disease. And typhoid fever is one of the health problems and endemic infection in Indonesia. This infection has increased every year by about 500 per 100,000 population, with a death rate of 0.6 until 5% in 2013. And then the incidence of typhoid fever is quite high, especially in the South Asia, Central Asia, and Southeast Asia. And based on the CDC, Center for Disease Control and Prevention, Indonesia, the prevalence of typhoid fever is about 358 until 110 per 100,000 people per year, including the incidence of typhoid fever in Islamic boarding school in Indonesia and in this city in Jombang. So how can the Salmonella typhi can be transmitted through the human? Because most of the people in Indonesia, especially in the boarding school, they buy a lot of food, street food especially, and we know that there is a lot of factors that can hit into the food. For example, the Musca domestica is a fly that can take into one food and others, and then go to garbage, and then to hand of people. So there's a lot of possibilities that factors that bring the bacteria, in this case is Salmonella typhi, can bring the disease for the student, especially in the boarding school. And then in this research, I bring two samples. The first samples is unwrapped and another is wrapped. It means that is there any difference between one with a wrapped food and another, because the mechanical picture of the Salmonella species is based on the fly. So I would like to analyze, is there any difference in this method? Okay, and the research type and samples. The type and design and research. The study uses a descriptive analytical research design so that it can identify and analyze the description of the Salmonella sp on the open or closed snack, or in this case is unwrapped and wrapped snacks at Islamic boarding school. And the population in the study is used one population, namely all the open and closed or unwrapped and wrapped snacks food sold in the Darul Islamic boarding school Jombang. And the sample size, the research took 5 samples from the open snacks or the unwrapped snacks, and 5 samples of closed snacks or wrapped snacks at Islamic boarding school in Jombang. And we considered the criteria of these samples. First is a fresh foods, so we avoid another transmission bacteria, because when we decided to the fresh food, so we can go to the focus on the bacteria at that time. Okay, and this is a research framework from the unwrapped and wrapped food sold in the area of Islamic boarding school Jombang, sampled by purposive sampling method according to inclusion and exclusion criteria. And then after that, the sample size 5 sample of unwrapped food and 5 sample of wrapped food, and we execute the microbiological tests and data processing, data analysis as a descriptive study, and the research result, as well as conclusion and suggestions. And location and time of research, the sampling stage was carried out in the Islamic boarding school area, and identification in the microbiological laboratory of the Dr. Thomas Surabaya. And this time of research is 14 March to 2019. And then the research variable is the independence variable in the study is contamination of Salmonella. And how the work algorithm of this research. The first is sample. We took a sample and then we bring to the hospital related to the microbiological test. And then we took a 1 gram aseptically, and then we give to the 25 milliliter MH broth and homogenize with vertex. And then we incubate in 35 degrees Celsius for 4 hours. And then after that, we will see how the bacteria that grow in the agar. Agar is place that we can see how many bacteria and how the bacteria grows with specifically. And then this we can see there is two colonies in here. The first is colorless and another is colorless. So it's really different between one bacteria and others. They can bring another colors based on the type of the bacteria itself. And then it is a microbiological test. And other hand, we have a biochemical test. So there's the SEA, there's SEM, and then Simon citrates. So this is how to analyze and distinguish between Salmonella sp and another. For example, Klebsiella pneumoniae, Escherichia coli, and another Enterobacteriaceae. And the result and the discussion, the population overview. The population for unwrapped is 5. It means that 50%, and wrapped is 50% as well. So the total is 100%. And then the result of the identification of Salmonella sp. Fortunately, for all the sample negative result of the Salmonella typhi. So 100% from the 10 food, whether unwrapped and wrapped food is 0 for the contamination of the Salmonella sp. And how about another identification bacterial contamination? So surprisingly, there are a lot of bacteria that contaminate in this food. Yeah, we know that in this earth we will have a lot of contamination with the bacteria, but we have to consider the amount of the bacteria itself. Is it safe or not with the amount of the bacteria at that time at the food? So surprisingly, there are a lot of number colonies or CFU that really high in certain foods, including in whether in unwrapped and wrapped foods. Not only just Escherichia coli, as we know, is a bacteria that also a normal bacteria that live in our body, but also Klebsiella pneumoniae. Klebsiella pneumoniae is a bacteria that oftenly live in the hospital, or we call it a nosocomial bacteria. So it is really rare that there is a Klebsiella pneumoniae in the food. So we need another investigation why there is this bacteria at the food. And this is the picture or the image of the our research based on the microbiological test. This is mechanical agar. And then this is a TSEA test, SIM test, and citrate test. It is a biochemical test. It's also how to distinguish between one and others is the color. Yeah, we can see there is a blue one, and there is an orange one, and there is a colorless one. So we have a criteria based on the bacteria live in the sample itself. So analysis of the for bacterial contamination on food. The number of typhoid fever incidents at 14 days after checking food. From the data obtained, there are 7 cases of typhoid fever in Darul Islamic boarding school. In a similar study, which took sample from street food, in the area of SD Pertiwi, Taman Sari Village, Bandung City, on 15 samples of snacks that had been tested, no contamination was found from Salmonella. And then the number of typhoid fever incidents at 14 days after taking food. From the data obtained, there are 7 cases of typhoid fever in Darul Islamic boarding school, and based on the research study, most 2016, 66% of samples collected in the morning were contaminated by Klebsiella pneumoniae, whereas 34% at night. This has something in common with this study because the researchers took all the food in the morning. The result, researchers found that 80% of the snacks cited were contaminated with the Klebsiella pneumoniae bacteria. And Min from Union Agriculture University in 2016 revealed that leaves plants in the field can be a reservoir for Klebsiella pneumoniae. And Escherichia coli contamination of food comes from almost exclusively from feces, and this is transmitted through contamination of food and water feces, as well as cross-contamination or through direct human contact during food preparation. So we have to see not only just from how they wrap, but also how they process or they're making the food itself, because it is probably they have no good preparation for handling and then for hygiene, etc. So we have to a lot of consideration from this bacterial contamination. And the conclusion, first, from the incidents, identification of 500 food, out of 5% or 0% were contaminated with Salmonella sp, which means that 500 food studied were not contaminated by Salmonella sp. And second, according to the identification of 500 food, 0 out of 5% were contaminated with Salmonella sp, which means that 500 food studied were not contaminated by Salmonella sp bacteria. And there are other bacterial contamination in unwrapped food that are 30% or 3 Escherichia coli and 4 Klebsiella pneumoniae or 40%. In 5 wrapped food, there were 3 samples contaminated by Escherichia coli or 30% and 4 samples contaminated by Klebsiella pneumoniae or 40%. Fala, so thank you. Any question or any suggestion or etc.? We're happy to listen to your advice. Thank you.
Thank you very much, Mohammed Zulfikar, for your presentation.